Hello and welcome to this channel. Today I want to show you how to use a dialysis tube to change the dialysis solution of your sample into a different solution. And here I'm pouring my second solution into a flask. Now I'm cutting some dialysis tubes. So the most important thing that you have to make sure is that the size of the molecular weight cutoff for the tube is smaller than the size of the protein that you have. So for me, my protein is 12 kilodalton and the molecular weight cutoff of this tube is 3.5 kilodalton. And this makes sure that your protein won't escape from the tube, so you won't lose your protein which you worked really hard for. And yeah, basically you need to hydrate it for a few minutes, then you will be able to open one side and the other side needs to be clipped, so you can easily pipette in your solution into the tubing. This process needs a little bit of patience, and you need to do use a pipette or any sort of pipette to do this. And yeah, after you pipette everything in, you can see it's stable inside. I'm gonna clip the other side and that's it. So thank you so much.